Step by step, they challenge, explore, and unlock the unknown. Fueled by imagination, alone and with others. Repeating an experiment 1,000 times to have the chance to get it right. Once, but for forever. Making brave choices, choosing to push the boundaries, and choosing again and again to be led by science. In everyday decisions, everyday interactions, no matter where they are. Because putting science at the center of everything is the best way to understand and transform disease. The best way to change people's lives. This is why we believe in what science can do. This is for scientists and those who believe in the potential of science. It starts with curiosity, a surprise in the pattern, an obstacle that can't be explained. Step by step, they challenge, explore, and unlock the unknown. Fueled by imagination, alone and with others. Repeating an experiment 1,000 times to have the chance to get it right. Once, but for forever. Making brave choices, choosing to push the boundaries, and choosing again and again to be led by science. In everyday decisions, everyday interactions, no matter where they are. Because putting science at the center of everything is the best way to understand and transform disease. The best way to change people's lives. This is why we believe in what science can do. This is for scientists and those who believe in the potential of science. It starts with curiosity, a surprise in the pattern, an obstacle that can't be explained. Step by step, they challenge, explore, and unlock the unknown. Fueled by imagination, alone and with others. Repeating an experiment 1,000 times to have the chance to get it right. Once, but for forever. Making brave choices, choosing to push the boundaries, and choosing again and again to be led by science. In everyday decisions, everyday interactions, no matter where they are. Because putting science at the center of everything is the best way to understand and transform disease. The best way to change people's lives. This is why we believe in what science can do. This is for scientists and those who believe in the potential of science. It starts with curiosity, a surprise in the pattern, an obstacle that can't be explained. Step by step, they challenge, explore, and unlock the unknown. Fueled by imagination, alone and with others. Repeating an experiment 1,000 times to have the chance to get it right. Once, but for forever. Making brave choices, choosing to push the boundaries, and choosing again and again to be led by science. In everyday decisions, everyday interactions, no matter where they are. Because putting science at the center of everything is the best way to understand and transform disease. The the best way to change people's lives. This is why we believe in what science can do. This is for scientists and those who believe in the potential of science. It starts with curiosity, a surprise in the pattern, an obstacle that can't be explained. Step by step by step, they challenge, explore, and unlock the unknown, fueled by imagination, alone and with others, repeating an experiment 1,000 times to have the chance to get it right, once, but for forever. Making brave choices, choosing to push the boundaries, and choosing again and again to be led by science, in everyday decisions, everyday interactions, no matter where they are. Because putting science at the center of everything is the best way to understand and transform disease, the best way to change people's lives. This is why we believe in what science can do. This is for scientists and those who believe in the potential of science. It starts with curiosity, a surprise in the pattern, an obstacle that can't be explained. Step by step by step, they challenge, explore, and unlock the unknown, fueled by imagination, alone and with others, repeating an experiment 1,000 times to have the chance to get it right, once, but for forever. Making brave choices, choosing to push the boundaries, and choosing again and again to be led by science, in everyday decisions, everyday interactions, no matter where they are. Because putting science at the center of everything is the best way to understand and transform disease, the best way to change people's lives. This is why we believe in what science can do. This is for scientists and those who believe in the potential of science. It starts with curiosity, a surprise in the pattern, an obstacle that can't be explained. Step by step by step, they challenge, explore, and unlock the unknown, fueled by imagination, alone and with others, repeating an experiment 1,000 times to have the chance to get it right, once, but for forever. Making brave choices, choosing to push the boundaries, and choosing again and again to be led by science, in everyday decisions, everyday interactions, no matter where they are. Because putting science at the center of everything is the best way to understand and transform disease, the best way to change people's lives. This is why we believe in what science can do. This is for scientists and those who believe in the potential of science. It starts with curiosity, a surprise in the pattern, an obstacle that can't be explained. Step by step by step, they challenge, explore, and unlock the unknown. Fueled by imagination, alone and with others. Repeating an experiment 1,000 times to have the chance to get it right. Once, but for forever. Making brave choices, choosing to push the boundaries, and choosing again and again to be led by science. In everyday decisions, everyday interactions, no matter where they are. Because putting science at the center of everything is the best way to understand and transform disease. The best way to change people's lives. This is why we believe in what science can do. This is for scientists and those who believe in the potential of science. It starts with curiosity, a surprise in the pattern, an obstacle that can't be explained. Step by step by step, they challenge, explore, and unlock the unknown, fueled by imagination, alone and with others. Repeating an experiment 1,000 times to have the chance to get it right. Once, but for forever. Making brave choices, choosing to push the boundaries, and choosing again and again to be led by science. In everyday decisions, everyday interactions, no matter where they are. Because putting science at the center of everything is the best way to understand and transform disease. The best way to change people's lives. This is why we believe in what science can do. This is for scientists and those who believe in the potential of science. It starts with curiosity, a surprise in the pattern, an obstacle that can't be explained. Step by step by step, they challenge, explore, and unlock the unknown, fueled by imagination, alone and with others. Repeating an experiment 1,000 times to have the chance to get it right. Once, but for forever. Making brave choices, choosing to push the boundaries, and choosing again and again to be led by science. In everyday decisions, everyday interactions, no matter where they are. Because putting science at the center of everything is the best way to understand and transform disease. The best way to change people's lives. This is why we believe in what science can do. This is for scientists and those who believe in the potential of science. It starts with curiosity, a surprise in the pattern, an obstacle that can't be explained. Step by step by step, they challenge, explore, and unlock the unknown, fueled by imagination, alone and with others. Repeating an experiment 1,000 times to have the chance to get it right. Once, but for forever. Making brave choices, choosing to push the boundaries, and choosing again and again to be led by science. In everyday decisions, everyday interactions, no matter where they are. Because putting science at the center of everything is the best way to understand and transform disease. The best way to change people's lives. This is why we believe in what science can do. This is for scientists and those who believe in the potential of science. It starts with curiosity, a surprise in the pattern, an obstacle that can't be explained. Step by step by step, they challenge, explore, and unlock the unknown, fueled by imagination, alone and with others.
Senhoras e senhores, bom dia. Solicito a todos que ocupem os seus lugares e aos portadores de telefone celular a gentileza de desligarem os seus aparelhos. Obrigada. Esta cerimônia será transmitida nos auditórios 1 e 2 desta faculdade no original. O cerimonial da Universidade de Brasília tem o prazer e a honra em dar início à solenidade de outorga de título de doutor honoris causa ao professor Bruce Alan Butler. E como parte da comemoração dos 50 anos do curso de Medicina da Universidade de Brasília. O título de doutor honoris causa foi concedido por meio da resolução do Conselho Universitário número 20 de 2015, de 13 de outubro de 2015. O título é concedido à personalidade que tenha se distinguido pelo saber e pelo conhecimento das artes, da ciência, da filosofia, das letras ou do melhor entendimento das coisas. Gostaríamos de agradecer aos extremos presentes, a decana de extensão da Universidade do Brasil, a professora Fernanda de Martins, da decana de Investigações Pessoais da Universidade do Brasil, a professora Maria Angela diretor da Fundação de Ensino e Pesquisa em Ciências da Saúde, senhor Armando Raio, representando o Conselho Regional de Medicina do Distrito Federal, a conselheira Adília Jane de Alcântara Seguro, e representando as Faculdades Integradas da União Educacional do Planalto Central, o coordenador do curso de Medicina, professor Marcelo de Souza Genari, e as demais autoridades aqui presentes. Para compor a mesa de honra, convidamos diretor científico do Nobel Media, senhor Adam Smith. Diretor médico da AstraZeneca Brasil, senhor Paulo Miranda. Vice-reitora da Universidade de Brasília, professora Sônia Nair Bal. Presidente do Conselho Universitário e reitor da Universidade de Brasília, magnífico reitor, professor Ivan Marques de Toledo Camargo. Neste momento, convidamos os membros da comitiva que recepcionará a mesa de honra ao homenageado, professor Bruce Alan Butler. Diretor da Faculdade de Medicina da Universidade de Brasília, professor Paulo César de Jesus. Diretora da Faculdade de Saúde da Universidade de Brasília, professora Maria Fátima de Souza. Diretora da Faculdade UnB Ceilândia, professora Diana Lúcia Moura Pinho. Diretora do Instituto de Ciências Biológicas da Universidade de Brasília, professora Andréia Queiroz Maranhão. Diretor da Faculdade de Comunicação da Universidade de Brasília, professor Fernando Oliveira Paulino. Coordenador de pós-graduação da Faculdade de Medicina da Universidade de Brasília, professor Leopoldo Luiz dos Santos Neto. Solicito aos convidados para receberem de pé e com aplausos o homenageado professor Bruce Alan Butler, acompanhado do decano de pós-graduação, professor Jaime Martins de Santana. Permaneçam de pé, ouviremos a execução do hino nacional dos Estados Unidos, seguido do hino nacional brasileiro.
És bela, és forte, padre do colosso, e o teu futuro noite berço esplêndido, ao som do mar e a luz do céu profundo. Fulguras ao Brasil foram da América, iluminado o sol do novo mundo. Do que a terra mais garrida, Deus de sonhos lindos campos tem mais flores. Nossos bosques têm mais vida, nossa vida no teu seio mais amores. Ó pátria amada, idolatrada, salve, salve. Brasil de amor eterno seja símbolo, volar os teus tempos estrelado. Digo verde louro desta flâmula, paz no futuro e glória do passado. Mas serve da justiça brava forte, verás que o filho teu não foge à luta, nem tem quem te adora contra a morte. Terra adorada, entre outras mil és do Brasil, ó pátria amada, dois filhos destes solos mais, pátria amada, Brasil.
Passamos a palavra ao presidente do Conselho Universitário e reitor da Universidade de Brasília, professor Ivan Marques de Toledo Camargo, para declarar a abertura desta sessão solene.
Muito bom dia. Declaro aberta essa sessão solene de outorga de título de doutor honoris causa ao professor Bruce Alan Butler.
Para um pronunciamento, passamos a palavra ao diretor científico do Nobel Media, Sr. Adam Smith.
Thank you so much. So I'm Adam Smith from Nobel Media, which is the media arm of the Nobel Foundation based in Stockholm, Sweden. And of course, the Nobel Foundation is the organization that awards the Nobel Prize annually. And on behalf of all at the Nobel Foundation and Nobel Media, I'd like to welcome you to today's lecture and ceremony. This lecture that will follow is part of the Nobel Prize Inspiration Initiative, and this initiative is a collaboration between AstraZeneca and Nobel Media, designed to take Nobel laureates around the world to meet audiences of scientists, especially audiences of young scientists, such as I see before me. The initiative has been running for 5 years, and we've been to many countries. We've been to Korea, China, Russia, India, Denmark, Sweden, the UK, the US, and I'm very happy to say twice to Brazil. And indeed, we've been here to the University of Brasilia before. And I'd like to extend a special thanks to the University of Brasilia for welcoming us again, and to Dean Camargo and Professor de Santana. Thank you so much for having us back. We're delighted to be here. Now, one of the things that I like to show our colleagues back in Stockholm is what this initiative is all about. And what this initiative is all about is having Nobel laureates talk to audiences such as I see in front of me. So if it's okay, I'd like to take a photograph of you all to send back to Stockholm. So you'll have to bear with me while I do this. It won't take a second, but it's quite important. Oh, no, hang on. We have to do a panel. Here we go. Here we go. Say cheese. Hello. Perfect. Thank you very much. Okay, I have very little more to say except to thank Professor Bruce Beutler in particular for taking the time 3 days out of his schedule to be here in Brazil. He's incredibly busy. As you'll hear, he has an enormous lab and works very hard, and it's lovely that he takes the time to be part of this initiative. And so I'd just like to express our special thanks to him. And that's all I have to say. Thank you.
Para proferir o discurso de apresentação do homenageado professor Bruce Alan Butler, convidamos o decano de pesquisa e pós-graduação da Universidade de Brasília, professor Jaime Martins de Santana.
Ilustríssimo senhor Bruce Beutler, laureado com o Nobel de Fisiologia ou Medicina de 2011. Magnífico reitor da Universidade de Brasília, professor Ivan Camargo. Senhora vice-reitora da Universidade de Brasília, professora Sônia Baal. Senhor Adam Smith, da Nobel Media, muito prazer em vê-lo aqui de novo. Eu espero que isso se repita muitas e muitas vezes. Muito obrigado. O senhor Paulo Miranda, diretor da AstraZeneca no Brasil, muito obrigado. Senhoras e senhores professores, distintas e distintos estudantes, autoridades, amigos da ciência da Universidade de Brasília, senhoras e senhores. Imensa alegria e grande emoção permeiam essa manhã na Universidade de Brasília, que recebe a visita do ilustre professor Bruce Beutler. Nasceu em Chicago, Illinois, no dia 29 de dezembro de 1957. Seus avós emigraram da Europa para os Estados Unidos, evitando perseguição violenta aos judeus. Cresceu ao lado dos irmãos e sob a atenção e carinho da avó materna. Os avós paternos eram médicos. Ele, clínico geral, e ela, pediatra. Saíram de Berlim em 1935 por causa do nazismo. Deixaram o ambiente social e politicamente hostil e chegaram à América em busca de paz e de educação para os filhos. A avó Beutler, carinhosa e severa, costumava dizer: Bruce, quem sabe você ganhará um Nobel um dia. Nesta foto, próxima foto, por favor. Não, próxima, vai errado. Próxima. Ok. Nesta foto, vocês podem ver os pais do professor Beutler, ele próprio, Bruce, a avó materna e os irmãos, por volta do ano 62 do século passado, na Califórnia. Próximo, por favor. Cresceu nesta casa. Percebe-se uma bananeira, eucalipto, coisas que tem no Brasil também. E interagindo naquela região com a rica natureza californiana, incluindo atenção especial aos animais e alguma indisciplina com o calendário escolar. Foi então que fez sua iniciação científica antes mesmo de começar a universidade, no laboratório do seu próprio pai e primeiro orientador. Quis saber se bactéria expressava glutationa peroxidase, uma enzima, e se isso estava sob a regulação gênica nos moldes daquilo que descreveram Jacob e Monod, alguns anos antes daquela época, também laureados. Os resultados foram completamente inesperados e intrigantes. Não havia enzima, mas havia uma catarse. Neste ambiente de pesquisa em bioquímica e biologia celular, Bruce apaixonou-se pela ciência. Resolveu então estagiar no laboratório do Dr. Dan Lindsay. Próximo, por favor. Esse é à esquerda. Um especialista e conhecido pesquisador de genética de drosófilas, iniciando-se no mundo da genética molecular, que naquela época estava nascendo. No verão que fez 18 anos, entrou em contato com um termo novo, endotoxina. Os mais antigos, como eu, se lembram desse nome. Os mais novos, não. Também conhecido como LPS, olipopolissacarídeo, uma substância da parede de bactérias. E como esta molécula ativava leucócitos. Mas esse assunto foi tangencial. E a pergunta: haveria um receptor de LPS na superfície de leucócitos que possibilitava a sua ativação? Essa pergunta não foi feita. Exatamente o que lhe daria o Nobel mais tarde. A pergunta não foi feita naquela época, mas foi feita mais tarde. Igualmente importante na formação acadêmica do Dr. Beutler foi a experiência de trabalhar no laboratório do Dr. Suzumo Ono, um famoso geneticista e grande teórico da biologia. Nesta época, o Dr. Ono resolveu aprender imunologia. E o jovem Bruce aprendeu com ele. E foi apresentado ao intrigante e fascinante mundo da defesa imunológica contra patógenos. Ainda aos 18 anos, com experiência em pesquisa laboratorial e um trabalho publicado já na prestigiosa revista Cell, Bruce estava confiante que seria aceito como estudante de medicina em todas as boas universidades americanas. Todas. Quem é que não queria um estudante jovem, com experiência em pesquisa e com uma publicação na Cell? Candidatou-se a várias delas, que nada, só a Universidade de Chicago aceitou. Muito provavelmente, as universidades não viram aquele jovem como futuro clínico, mas como um cientista. Gostou de Chicago, mas como californiano, igualmente brasileiro, sofreu com o frio e o vento, trabalhou forte e adorou as disciplinas, principalmente as básicas, como fisiologia e microbiologia. Chegada a hora de aprender clínica médica, no entanto, sentiu falta do trabalho de pesquisa em laboratório. Graduou-se em 1981 e, seguindo o conselho do pai, foi para a residência médica. Escolheu então o melhor lugar à época, Dallas, onde exigiam-se independência e iniciativa dos profissionais. O objetivo do pai era garantir o futuro do filho como clínico, caso a pesquisa não prosperasse. Acho que os pais continuam com essa preocupação. Talvez seja uma característica genética. E foi então que o jovem Bruce Boydler descobriu definitivamente que a prática médica não era para ele. A saudade da pesquisa laboratorial só aumentava. Tinha o sentimento de conhecer as proteínas, de saber como estudá-las, de isolá-las, coisa que aprendeu no laboratório do pai, ainda muito jovem, e de estudá-las no contexto da imunologia. Desta forma, partiu com a esposa e o filho mais velho para Nova York e foi trabalhar na Universidade Rockefeller. Lá, após meses de trabalho duro, descobriu o fator de necrose tumoral, o TNF, em camundongos. Mais importante do que isso, descobriu suas propriedades inflamatórias. Para que isso se fizesse, o Dr. Boydler passou pelas dificuldades experimentais ordinárias daquela época, como isolar uma proteína, o soro, e identificá-la molecularmente. Essa, nesse caso, estava com o N terminal bloqueado. Ai, meu Deus, para fazer sequência de peptídeo, muito difícil. Várias publicações importantes e reconhecimento profissional internacional não foram suficientes para fixarem o Dr. Boydler em Nova York. Foi para o Texas, mais precisamente para Southwestern Medical Center, UT, procurar autonomia como pesquisador e construir seu futuro. Este futuro que o Dr. Boydler irá nos contar hoje. O trabalho do Dr. Boydler e sua equipe foram de grande importância para sabermos como o TNF é produzido, seu papel na inflamação, como macrófagos e células dendríticas reconhecem patógenos. Próximo, por favor. E são ativadas por eles. Quais os receptores envolvidos, ou quais os receptores envolvidos, e, enfim, como se dá a regulação da inflamação e da resposta imune inata e adaptativa. E muito importante, como que uma molécula, aqui embaixo, na superfície da célula dendrítica do macrófago, reconhece patógenos, ativa-se e ativa o sistema imune adaptativo e faz uma resposta imune. Muito importante, porque desde a descoberta de que micro-organismos causavam doenças, não se tinha chegado a um conhecimento tão importante de que como era o início da defesa contra esses patógenos causadores dessas doenças. Por isso, Beutler e colegas ganharam o Nobel de Fisiologia ou Medicina de 2011. Importante salientar aqui que esse conhecimento resultou em medicamento anti-inflamatório que alivia a dor e o sofrimento de milhões de pessoas em todo o mundo, que possuem diversos tipos de doenças com essas características. É a pesquisa básica, fundamental, que alimenta a pesquisa aplicada, a tecnologia e a inovação. A visita de um laureado Nobel ao UnB é importante porque temos a oportunidade de cultivar a ciência que avança de forma contundente e qualificada, com implicações positivas para a compreensão de como a vida e toda a natureza funcionam. É um momento de profunda reflexão sobre o papel do cientista na formação de valores que são pilares para uma sociedade cada vez mais serena, em busca da paz e da compreensão da diversidade cultural. Para os estudantes trata-se de uma aula absolutamente única na universidade. Saber qual foi o caminho, com a sua dificuldade e alegria, que o Nobel trilhou. Os estudantes têm a chance de sentirem que um Nobel é um ser humano comum, mas que tem uma curiosidade científica intensa. De saberem da fonte como as perguntas científicas são pensadas e as buscas das respostas são trabalhadas com grande entusiasmo e dedicação. É um exemplo de vida profissional dedicado à compreensão de como a natureza acontece. E de como a ciência colabora para a preservação da vida e a melhoria de sua qualidade. Muito obrigado, Dr. Beutler.
Para um pronunciamento, passamos a palavra ao diretor médico da AstraZeneca Brasil, senhor Paulo Miranda.
Bom dia a todos. Em primeiro lugar, não vou resistir, mas uau! Para mim, assim, a primeira coisa que a universidade traz para a gente é a sensação de se sentir jovem de novo. E essa é a razão pela qual eu acredito bastante no meu trabalho e no que a gente está fazendo. Eu estou fazendo um leve desvio de protocolo, porque o Adam me deu essa liberdade a fazer uma panorâmica de todos vocês. Mas eu queria começar dizendo isso. O quão importante, o quão significativo é para mim e para a empresa, para a AstraZeneca, poder trazer esse tipo de inspiração para todos vocês. Eu queria começar primeiro agradecendo à Universidade de Brasília por ceder esse espaço, por criar essa oportunidade, em particular ao magnífico reitor, professor Ivan Camargo, ao professor Jaime Santana e à vice-reitora Sônia Bal, de aceitarem essa iniciativa para trazer um prêmio Nobel aqui dentro da Universidade de Brasília. Eu gostaria também de agradecer ao professor, ao Dr. Roland Beutler, pela presença dele, pela disponibilidade de vir ao Brasil, e ao Nobel Media por essa parceria com a AstraZeneca nesse tipo de atividade. Por fim, eu gostaria realmente, sinceramente, de agradecer a presença de todos vocês aqui. Eu sei que tem mais 2 outros auditórios cheios, e é isso que faz para a gente valer o dia, valer o ano, porque é aquilo que a gente acredita. A AstraZeneca tem como um dos seus pilares a liderança científica nas áreas terapêuticas que a gente atua. Um dos nossos valores nós perseguimos, nós buscamos o desenvolvimento científico, e vocês viram no vídeo que passou antes que o slogan da AstraZeneca é What Science Can Do, o poder da ciência. Nós realmente acreditamos que é através da ciência que a gente vai conseguir trazer inovação e trazer melhoria para toda a sociedade. Como parte disso, a AstraZeneca também tem outras iniciativas nesse sentido, como uma parceria com o CAPES e o CNPq, através do projeto do programa Ciências Sem Fronteiras, para levar estudantes de pós-doutorados para as unidades de pesquisa em biológicos da AstraZeneca na Inglaterra e nos Estados Unidos. E hoje nós temos 30 bolsas de pós-doutorados oferecidas, nós já temos 12 bolsas a 12 estudantes de pós-doutorados na Inglaterra e nos Estados Unidos, aproveitando essa oportunidade de trabalhar e conduzir suas pesquisas em laboratórios avançados. Além disso, a AstraZeneca tem uma parceria, uma colaboração que assinou esse ano com a FAPESP, a Fundação de Amparo à Pesquisa de São Paulo, do Estado de São Paulo, para a pesquisa básica em doenças cardiovasculares e metabólicas. A gente piamente acredita que, através desse tipo de colaboração, a gente vai estar contribuindo para o desenvolvimento da ciência, para o desenvolvimento da pesquisa e contribuindo para trazer a inovação para o Brasil, que é uma das prioridades e uma das necessidades que a gente tem. Por fim, eu queria, uma vez mais, agradecer a presença de todos vocês, dizer que, para mim, é uma honra, isso faz valer a opção que eu fiz de carreira como médico, como hematologista, que estudou em livros de medicina escritos pelo pai do Dr. Beutler, de poder trazer essa oportunidade para todos vocês, e eu espero que todos vocês aproveitem essa palestra e essa oportunidade como realmente um momento inspirador para a carreira de vocês dentro da ciência. Muito obrigado.
Neste momento, o presidente do Conselho Universitário e reitor da Universidade de Brasília, professor Ivan Marques de Toledo Camargo, concede o título de doutor honoris causa ao professor Bruce Alan Butler e, em seguida, veste a epitoche e entrega o diploma.
Eu, Ivan Marques de Toledo Camargo, presidente do Conselho Universitário e reitor da Universidade de Brasília, confiro ao professor Bruce Alan Butler o título de doutor honoris causa que lhe foi concedido pelo Conselho Universitário, de acordo com o que preceitua o Estatuto da Universidade de Brasília.
Excuse me.
Our symbol.
Symbol.
Thank you. Thank you so much.
Para um momento musical, convidamos o professor Ebenezer Nogueira no fagote e o Sr. Lucas Donato no violão.
It's Shaolin. It's a very Brazilian music. Okay?
I see.
I hope you don't need translator now.
No, okay. This is really nice. Thank you. It's really very beautiful.
Yes, it's nice. Yes, it's nice.
Excuse me, but I have to sign.
You have to sign it. Okay, it's better to sign.
Sure.
Okay.
This one?
Thank you.
it's silent.
But to make sure we have people to help us with microphones.
Nosso Nobel deve estar estreando fagote e violão. O nosso Nobel deve estar estreando fagote e violão aqui, né? Mas ele tem que entender que é bem comum aqui no Brasil a gente tocar fagote e violão em todas as esquinas, tocando peças brasileiras. Não é como nos Estados Unidos, né? Bassoon and guitar. Nós vamos tocar uma peça do Valdir de Azevedo, André de Sapato Novo. André and his new shoes.
Okay. Beautiful. Yeah. Wow. Thank you.
Ouviremos ao pronunciamento do magnífico reitor da Universidade de Brasília, professor Ivan Marques de Toledo Camargo, que em seguida irá declarar o encerramento desta sessão solene.
Alô, meu caro homenageado, professor Bruce Allen Butler, professora Sônia, professor Jaime, caro decano, professor Paulo, prazer tê-lo aqui. O Adam Smith já saiu, queria cumprimentar os nossos colegas da Leopoldo, Paulino, Andréia, Diana, Fátima e Paulo, todos os demais presentes, e terei que ser muito breve. Né? Quem vai falar hoje é o nosso homenageado, é o professor Prêmio Nobel de Medicina. O tempo de todos aqui é para ouvi-lo, mas eu não posso não agradecer nem a Nobel Media e a AstraZeneca pela trazer essa oportunidade para a Universidade de Brasília de ouvir o Prêmio Nobel. Né? A gente acha que isso é normal e não, né? Ter a honra de ter um grande mestre, um grande cientista, um grande acadêmico na nossa universidade é sempre uma honra. E como Paulo disse, quando estamos na universidade, a gente sempre se sente jovem. É muito bom estar nesse auditório lotado para ouvir um pouco da experiência do nosso professor, agora professor dessa universidade, professor doutor honoris causa da Universidade de Brasília, referência para todos nós, Bruce Allen Butler. Então, com essas palavras, eu declaro encerrada essa solenidade para dar início à solenidade mais importante, que é ouvir o nosso caro doutor, que vai fazer uma palestra para a gente. Muito obrigado, e obrigado pela presença de todos vocês. Está encerrada essa solenidade.
Nesse momento, a mesa de abertura se desfaz e convidamos a todos para ouvir a palestra Desconstruindo o Sistema Imunológico através da mutagênese germinativa aleatória, que será proferida pelo professor doutor honoris causa Bruce Allen Butler.
Thank you. So I am live now.
Também, é claro, né?
Well, thank you all very much. I want to start with a few words of gratitude for this great honor you've paid me. I have to start by saying that, as you've heard already, I was interested in nature from an early age. That was one of my defining characteristics when I was a child. And Brazil figured into my interest in nature from the start. I thought back about when I had first heard of Brazil as a country. It must have been when I was a very small child. I remember my older brother, Steve, was a stamp collector, and he convinced me to collect stamps as well. And back in the early 1960s, I remember having a beautiful series of stamps showing Brasilia with the green buildings that I've seen for the first time just earlier today. And it seemed an amazing thing to me then that a new city was being carved out of the wilderness. It was planned from the start, and it was to be a shining gem of the country of Brazil. Now, I learned about all of the wildlife of Brazil, of Amazonia, about the tremendous biodiversity, and I still love that today. I still have not been to the Amazon, but it's very high on my list. Interestingly, Brazil was the first country I ever visited outside the United States, with the exception of Mexico, which was just such a short drive from my house when we were growing up. When I was 14, my father came to an international hematology congress, and I was delighted that I got to come along. And I spent a week or so in Rio and another week in Sao Paulo. And then, as now, I thought the Brazilians were the most gracious, lovely, hospitable people on the planet. And so this is something that really means a great deal to me. Your university is young, and it has a pioneering spirit, I'm quite sure. 50 years is not very old for a university, and you're only a little bit older than that. Thank you. Only a little older than that. I, too, went to a young university. I went to the University of California at San Diego, which was only about 3 or 4 years old when I joined it. And there was an energy associated with that. And I'd like just to quote something else my father told me that I've never quoted before. He answered this statement that people often make in the United States and elsewhere: how can we compete against huge laboratories? How can we really do good work in science? Perhaps all the easy things have already been done. And he said, no, that's not true. The measure of a scientist is not determined by the size of his or her lab, but by the size of his or her ideas. And I think that's something that you can certainly capitalize upon here. One can still make great breakthroughs, great discoveries, and everyone should know that. No one should be afraid to try. I'll tell you a little bit more of my own personal story as I talk, and I'll tell you a story about genetics and how it's changed and how we have an exciting new approach to discovering the function of genes in the mouse. My story in science, the main one, goes back to the 19th century. I think all of you are familiar with Robert Koch, who's here in the foreground. He was one of the early microbe hunters. He established that there were certain rules for declaring that a microbe caused a certain infectious disease. Together with Pasteur, or perhaps we should say in competition with Pasteur, Robert Koch was one of the great microbe hunters and formed the germ theory of disease. Before him, people looked at infections as some mysterious act of God, and it's hard even to put oneself in that mindset. We're so familiar with the fact that germs cause disease. The man standing behind him is Richard Pfeiffer. He is much less well known, but he too did something extremely important. He came to Koch's laboratory in Berlin in 1891. he was assigned to work on cholera, which was one of the dread diseases in Europe at that time. And he determined that there was something intrinsically toxic about cholera vibrio, the organism that caused cholera. He could inject cholera in a heat-killed form into guinea pigs, and the guinea pigs would sicken and die looking like they had a real infection, where in fact they did not. He couldn't recover any living organisms from the peritoneal cavity. So there was something that was poisonous associated with bacteria, and he called this substance endotoxin to distinguish it from the proteinaceous exotoxins that had already been found. Some decades went by, and a great interest in endotoxin developed. It was discovered that it was associated with all gram-negative bacteria, and furthermore that it was part of the surface of gram-negative bacteria, the major lipid component of the outer leaflet of the outer membrane. It was an amphiphilic molecule that had a polysaccharide chain attached to some lipid chains that you see at the bottom, which we call lipid A. And lipid A was the toxic part. As late as the 1980s, people managed to synthesize artificially lipid A, and they found that it did everything that Pfeiffer had observed about 90 years earlier. This is how we might represent an LPS molecule today, with a short polysaccharide chain attached to a 6-acyl chain lipid A. Now, Richard Pfeiffer, as I said, is rather obscure. Many of you probably never heard of him, and yet during his lifetime he was very famous. He was nominated 33 times to receive the Nobel Prize in Physiology or Medicine, and he died in the mid-1940s. Why was this so? Well, first of all, thousands of people die every day of gram-negative septic shock around the world. Probably even more people died in Pfeiffer's time, despite the fact that the population was smaller. This is what gram-negative sepsis looks like. To those of you who aren't physicians or physicians in training, this is a baby with meningococcal sepsis. You see that there's spontaneous bleeding into the skin, formation of ecchymoses. You notice that the child is intubated because the lungs tend to fill with fluid, and there's a great deal of organ injury going on, especially to the kidneys, the liver, other structures as well. Long ago, in Pfeiffer's time, it was recognized that this was a severe acute systemic form of inflammation. And one has to recognize that until recently, the origins of all inflammation, whether infectious or sterile, were obscure. No one knew really what the trigger was. It was widely guessed that inflammation was a process that arose in evolution to help combat infection. Nearly 100 years went by between the identification and naming of endotoxin and the discovery of the receptor for lipopolysaccharide. And that receptor was the key to understanding how we become aware of infection, we mammals and we humans. I began to be interested in endotoxin about 40 years ago. And I'll have to back up just a little bit. I had a lot of encouragement from my father to not merely observe nature, but to learn something about science. And he recommended certain books to me, among them these two books, The Microbe Hunters by Paul de Kruijff and Aerosmith by Sinclair Lewis, who won the Nobel Prize for Literature, by the way, largely because of this book, and before that, the Pulitzer Prize for this book, which he declined. It was given to him for establishing a new style of writing, and he said, no, that wasn't good. People shouldn't try to emulate the new style. They should develop styles of their own. But anyway, I recommend both these books to all of you if you haven't read them. They inspired me to want to be a physician, to want to study disease, infectious disease especially, and to make discoveries like the great early microbe hunters. In the chapters of the Microbe Hunters, Paul de Kruijff covers not only Koch and Pasteur, but Spallanzani and Metchnikoff and Ehrlich and all the people that one subsequently learns about in medical school, and I wanted to be just like them. It happened that in 1975, while I was in college, I worked in the lab of this man, Abraham Braudy, who did work on gram-negative bacteria, on Brucella especially, and he also was a pioneer in endotoxin research. He worked to develop antibodies that might be used to neutralize endotoxin and prevent endotoxic shock. And that was never successful. However, it's undoubtedly from Abraham Braude that I first learned the word endotoxin and learned about LPS. When I was 18 or 19, my father encouraged me strongly to go to medical school as an avenue to being a biomedical researcher. He said that I should learn about anatomy, physiology, pharmacology, about how a whole organism works, and then I'd be well qualified to study any biomedical problem I wanted to. And he said also, as was mentioned, that if I didn't do well in research, I'd have a job to fall back on. I could always be a physician. And I think all of this was very good advice, and in particular, the first part of it was excellent advice, and I believe to this day that studying nature by studying disease, learning what goes wrong and therefore what has to go right, that's what's made medicine such a powerful force in teaching about biology. So I went to the University of Chicago, the only university that accepted me, and that was how I looked, because that was my admission photograph. And at that time I was 19, and I think I look exactly the same today. This is the University of Chicago. You can see the quadrangle here in the foreground and the tall skyscrapers of Chicago along the shore of Lake Michigan in the background. And that was how the school looked and how it still looks. And I spent the next 4 years there pretty much immersed in medicine, but always trying to work in various laboratories as well, because I loved working in the laboratory. I'd had the experience of working in my father's lab, the Braude lab, also Susumu Ono's lab. I worked in herpes virus labs here and in neurology labs. I loved the very environment of the lab, and I loved trying to solve problems, even if at that stage they were very small. That was kind of my scientific infancy. After I finished medical school, my father, who was always a good source of guidance to me, said, you should do at least a year of internship, and then you'll be a polished, finished doctor, and then you can decide what you want to do. And I did a year of internship in internal medicine. I went to the toughest program in the country to stress myself, because I had a feeling I wouldn't be doing it very long. And I did, in addition, a year of neurology. And I learned a lot more about infectious disease then. Of course, I'd been seeing it all along in medical school, but I perhaps learned quite a bit more about it from outstanding infectious disease doctors whom I've listed here. And interestingly, my brother as well, because he had preceded me in medicine, and he did specialize in infectious disease. And to this day, I often talk to him about the clinical phenotype of various infectious illnesses. After 2 years, I missed working in the lab so much that I decided this was the end of medicine for me in terms of medical practice, and I wanted to really start my career and work on something important. And I went to the Rockefeller University, where I became a postdoctoral fellow. This is in New York, and the tall white building over here is where I worked. This is the tower building on the Rockefeller University campus. This really does belong in the picture. This is a tramway to Roosevelt Island in the middle of the East River. The subject in that lab, which was headed by Anthony Cerami, had to do with wasting in chronic disease. Now, I'll tell you in advance that what we found finally had really rather little to do with wasting, but we were interested in the subject of cachexia. This is a cow with trypanosomiasis, not T. cruzi, but T. brucei brucei. And these animals typically become severely wasted as the result of infection. They might have quite a low pathogen burden, only a few grams of microbes in their blood and lymphoreticulae organs, and yet they stop eating, they lose a lot of weight, and they appear to be hypercatabolic. No way can one account for this by a competitive mechanism, because the mass of parasites is trivial, essentially, and it seemed likely that the trypanosomes must be sensed by cells of the immune system, perhaps the innate immune system, perhaps macrophages, and they would make a factor in turn that would act on energy storage tissues of the host in order to cause the catabolic state. We defined such an activity called cachectin, which was made by macrophage in small amounts in response to exposure to trypanosome organisms. And this factor would act on adipocytes to shut off lipoprotein lipase, acetyl-CoA carboxylase, and fatty acid synthetase, which are all enzymes needed either for the uptake of fat or for its de novo synthesis. There were other aspects to the whole hypothesis. We felt that certainly not only trypanosomes, but things made by mycobacteria or perhaps endotoxin might induce cachectin synthesis. We thought that perhaps tumors would do it as well, because cancer famously causes wasting, and perhaps under some circumstances cachectin might be made autonomously. As I looked at all aspects of the model, it became clear to me that endotoxin was by far the most potent inducer of a cachectin activity. And I therefore set out to purify cachectin to homogeneity and to try to sequence it using endotoxin as an inducing agent. I added endotoxin to cultures of immortalized mouse macrophages, and after 4 to 6 hours I would harvest the medium, working ultimately with hundreds of liters of medium that I would concentrate and then fractionate. I would add that in turn to 3T3L1 cells, and I'd look for the shutoff of lipoprotein lipase. That was the assay. I had some experience at purifying proteins from having worked in my father's lab. In fact, I'd even crystallized a protein just for practice, but not de novo. Nonetheless, I was pretty confident of my skills, and in the course of about a year, I purified cachectin to homogeneity, and I found that a single protein species comprising about 1 to 2% of the total protein made by macrophages after you activated them with LPS had all of the cachectin activity. And this is how that protein looked on a gel. It weighed 17,500 Daltons, and it was a very pure single band. I struggled with it for a while and had to repurify it by a different method, and finally I determined the N-terminal sequence, getting 17 amino acids, if I remember the number right, maybe a few more. And I thought for a time that this was a unique and new protein. One day, though, I got a phone call from a friend to whom I had given some purified cachectin, and he told me he happened to notice that it had very high tumor necrosis factor activity. Tumor necrosis factor of human origin had been isolated just recently by Genentech, and it was defined as a factor that would cause the death of tumor cells in culture or the hemorrhagic necrosis of tumors growing in mice. And I looked then at the human TNF sequence and found that it was strongly homologous to mouse cachectin, and I thought that quite clearly this was the mouse ortholog of human TNF that we had isolated, and that proved to be the case. This had me very excited. I felt that here we knew a whole different side to tumor necrosis factors activities. It wasn't simply the non-toxic agent that people envisioned for use in cancer therapy. It had toxicity, and if I thought about the matter, here this factor was made in abundance in response to LPS, and it did things that LPS could do in vivo, shutting off anabolic activities of the cells and also causing necrosis of tumors. And I began to wonder, could this be the factor that induces all of the effects of endotoxin? And I thought perhaps it was even responsible for the lethal effect of LPS. I purified quite a lot of natural material and later recombinant material, and I administered it to mice. When I gave 20 micrograms of this protein to a mouse, you see the animal on the right collapsed, had some diarrhea, has ruffled fur. Eventually it died as a result of being given TNF, where if I gave heat-activated TNF to another mouse, it looked quite healthy and really experienced no untoward effects. So definitely it was toxic, and to all outward appearances it looked like it might be a mediator of LPS effect. But this wasn't enough. Of course, in those days it was impossible to target genes. We're talking about 1985 here. And instead I raised an antibody against TNF, as we were then calling it. I injected this antibody into mice, and I then challenged them with LPS afterward. And this experiment was done with large numbers of mice in various different ways. This is only one experiment, but very consistently, passive immunization against TNF would lead to resistance to the lethal effect of LPS, but only partial resistance, indicating that there were other factors that contributed to the lethal effect. The dose lethality curve was shifted about threefold at the mean lethal dose. So by that time, we knew that LPS interacted with macrophages. It caused the production of TNF and some other cytokines. These would interact with receptors on other cells, and that would shape the inflammatory response that was created by LPS. You're already starting to wonder how the LPS works, I suppose. At least I did. I thought that's really a mystery. Nobody really knows the receptor for LPS after all this time. This was a little bit too early to begin tackling that problem, though. Instead, I began looking at other activities of TNF, and together with other researchers, found that in almost any cellular system, TNF caused inflammatory effects. It would activate neutrophils to bind to the endothelium of blood vessels, for example. It would activate fibroblasts to secrete collagenase, and it made sense that it might be involved in many inflammatory processes. At that point, I moved from Rockefeller and went to establish my own laboratory at Dallas, at UT Southwestern again, the second time I moved there, and I decided it would be really useful to have a completely non-immunogenic, completely specific inhibitor of TNF. And I designed one by hooking the extracellular domain of the TNF receptor, which was cloned in 1990, to an antibody heavy chain. And these were both endogenous components and wouldn't provoke an antibody response and were very specific for binding to TNF and neutralizing it. In fact, this is a mock-up of how the binding occurs based on the real interaction with the receptor to TNF. And we believe that the receptor occupies 2 of the 3 binding sites on the surface of this trimeric cytokine, and then it can no longer bind to the surface receptor and cross-link receptors on the surface. This ultimately was developed by Immunex and later Amgen, and it became known as the drug Enbrel, which was effective in treating rheumatoid arthritis and ankylosing spondylitis and other inflammatory diseases. But all the while I was wondering about how LPS worked, and that seemed a deeper scientific question to me. It seemed really the most important question in all of immunity and inflammation biology. Now, why did I think that? Well, I might have been really overestimating things, but it seemed to me that nobody understood how inflammation started. And nobody understood here now in the 1990s how the immune system perceived infection, which it does within minutes after microbes are inoculated. The receptors just weren't known. And LPS was known to be one of the things that's recognized, so why not track down the receptor and see what we could find? Something was known by 1990 about the LPS receptor. Wright and Ulevich had found that there was a surface protein called CD14, which had leucine-rich repeats in it, and which was necessary for LPS to signal, because antibodies against CD14 would completely block endotoxin responses. But there was no cytoplasmic domain to this receptor, and there must be some other molecule that would transduce the signal across the membrane of a macrophage, and nobody knew what that might be. Nor did anyone know exactly how it signaled. It was known, where TNF was concerned, that the transcription factor NF-kappa B would translocate to the nucleus and would drive transcription. Once the mRNA was made, it was in an untranslatable state, we found, but it could be turned on by a second signal that would allow the RNA to be translated and TNF to be produced and processed and secreted. Far and away, the big question was, what's this receptor and how do we perceive microbes? We tried to find the receptor using all the conventional approaches I knew of in those days. We used protein chemistry, immunochemistry. We used expression cDNA cloning, even insertional mutagenesis, and we worked at it for 3 years, and none of that worked. Ultimately, a pure genetic approach was used to find the receptor, and this is where I fell back on what I had learned from genetics teachers in the past. It had been known since 1965 that there were certain mice that you could give enormous doses of endotoxin to and they wouldn't respond at all. The first of these was C3H-HEJ, and that was the discovery of Hepner and Weiss, that these mice were refractory, apparently for genetic reasons. Later, it was found that if you give them real gram-negative bacteria, then they do die very quickly and very easily, which says that recognizing endotoxin is important to overcoming the infection. It's a signal that infection is going on. If your immune system remains ignorant of infection, it'll be overwhelmed eventually by the bacteria. In 1978, another strain unrelated to C3H-HEJ was discovered, C57 Black 10 SCCR. You see it looks different than the other mouse. This was the discovery of Coutinho and Mayo. And they crossed the two strains together and found that the offspring were also refractory to LPS. And this said that the same locus was hit, that we had two different alleles of the same gene that were defective. And in the same year, using classical markers, Watson and Riblet tracked down the chromosome for this gene to mouse chromosome 4, but only to a huge region of the chromosome. In those days, it wasn't possible to go much further. Beginning in 1993, as there got to be more than 100 molecular markers across the mouse chromosomes, my colleagues and I decided to use positional cloning to find the gene. Positional cloning means cloning by phenotype, and I'll just tell you that it is possible by looking at a trait in a mouse to track down the location of the gene wherein variation causes that trait. And this is done by defining a proximal and distal marker that bracket the mutation, cloning all of the mutations, all of the DNA between those two markers, and then looking for differences between an affected and unaffected strain. And this was very difficult in those days because methods for sequencing DNA were so difficult, and because nobody knew the location of any genes within the majority of the mouse genome. But we set out to do this, and we had 16 markers to work with on mouse chromosome 4. We set them all in order with respect to one another using genetics. We found the location of the LPS mutation, but only to a rather huge area. And even looking at 2,000 mayoses and finding new markers, we were only able to circumscribe the mutation to a 2.6 million base pairs in length. And of course, the mutation could be, and ultimately proved to be, only one nucleotide change. And sequencing was done by hand in those days with radioactive nucleotide incorporation. Nonetheless, we set about cloning all of the material across that region. These are bacterial artificial chromosomes containing mouse DNA. We had a large number of them, as you see, and there was one yeast artificial chromosome as well. And we started in the middle of our contig of overlapping pieces of DNA, and we sequenced in both directions toward the edges because it was most probable that the mutation would be in the middle. This went on for 3 years, where we would relentlessly fragment backs. We would sequence them, and we would inoculate new colonies from other backs during the night and grow them up and sequence them in turn and keep looking and looking using mostly computational methods to see what genes might be in the area. And we found only these pseudogenes in the mid of the contig. And of course, they weren't labeled as such. We had to painstakingly prove that they didn't code for real products and that there were no mutations distinguishing the affected from control strains. We were getting quite nervous because by 1998, more than 90% of the critical region had been covered, and it was always possible that we'd made a mistake and we were looking in the wrong region. The true mutation might be upstream or downstream. But we had invested too deeply in the project by that time, and even though we had covered almost all of it, I decided just to continue. Then, as always happens, you find something in the last place you look. And one evening I was in my study looking over the blast results, and I saw a very strong match between a database of RNA sequences and a putative gene in our contig. This was called the toll-like receptor 4 gene, and as I looked at it, I started to hyperventilate, and I got tachycardic, and I called Alexander Poltorak, the lead postdoc on the project, and tried to explain why this was so exciting, and he understood, and he started to hyperventilate too, and soon we could barely talk with each other. So why were we so excited by what we found? Well, remember I told you there was this molecule CD14 that was leucine-rich in the ectodomain, and the new molecule coded for a protein that also had a leucine-rich repeat ectodomain. And it was a transmembrane domain protein, excellent candidate to be the missing co-receptor. And we could imagine that by proximity or transfer of the ligand, it would bring LPS into contact with this new molecule and trigger a signaling event. More exciting, on the cytoplasmic side, this molecule looked a lot like the interleukin-1 receptor. The interleukin-1 receptor binds a protein, interleukin-1, which is known to have inflammatory effects. It's known to activate NF-kappa B. Therefore, our new receptor probably could activate NF-kappa B in some way. Also, I remembered having seen about 2 years earlier on the cover of Cell a paper about toll, the namesake of the whole protein family. And this was a paper written by Jules Hoffman and his colleagues, who had shown that in Drosophila, toll, which had been known as a developmental protein, had also an immunological role. In adult flies, if there were mutations in toll, the animals would become overgrown by fungal infection if you injected them with fungal spores. And this seemed a parallel circumstance to what we knew to be the case in our mutant mice. In mouse mutants, at the LPS locus, as we called it, if you inject with gram-negative bacteria, they would become overgrown by the infection. And so it could be, perhaps, that this was a conserved mechanism for recognizing microbes. Of course, we'd had good stories before. With all of the pseudogenes, we'd made up some plausible reason why each of them might be what we were after, and this would mean nothing if we couldn't find a mutation. But within a couple of days, we did find a mutation. We dropped everything else and began sequencing TLR4, and we found that there was a substitution of an A for a C within that part of the gene that coded for the cytoplasmic domain of the receptor. And specifically, there was an exchange of a proline, a conserved proline for a histidine, and it looked very much like it might damage the protein and prevent signaling. This wouldn't have been enough to publish, however, because people might have said, well, that's just happenstance. These strains have been separated for 35 years. What do you expect? And therefore, we had to go and look at the other strain that I told you about. We knew there was an allelic defect in C57 black 10 SCCR mice. There we found we couldn't amplify the cDNA at all, and we couldn't detect the messenger RNA on northern blots. It seemed to be gone, and within a few weeks we found that there was a deletion spanning 74 kilobases of DNA that cut out all of the coding region of the gene but affected no other genes, and this was a natural knockout of TLR4. So with that, we were quite certain that we had found the affected gene after 5 years of effort. We had good reason to believe that this was a true receptor for LPS, and I won't go into that. I'll tell you more the definitive result of Gio Li and colleagues, who 9 years later succeeded in crystallizing the complex of TLR4 together with another protein that was identified the next year in 1999 by Kensuke Miyake and colleagues. It's shown in magenta, and cyan is the color of the TLR4. You can see that the magenta MD2 molecule, as it's called, is a kind of a basket, and the acyl chains of LPS fit into that basket, and they also bind directly to the opposite, if I can call it that, the opposite chain of TLR4, and they cause a conformational change that's noticeable and well-defined now, and that is transduced across the membrane, and that activates the LPS response. So everything you saw in that baby with meningococcal sepsis happens right here with this molecule. This is how that intense inflammatory response is generated. And we know in a mouse that only about 0.1 nanograms of this complex is enough to render the lethal effect of LPS. That first night, we knew that there were other TLRs as well. We know now there are 10 in humans and 12 in the mouse, and they each detect a different signature molecule from microbes. TLR2 detects lipopeptides, TLR9 detects single-stranded DNA, TLR3 detects double-stranded RNA, and a lot of the crystal structures are now known. And collectively, you can imagine, all of these receptors can detect almost any microbe that you can ever encounter. And indeed they do. And if you don't have any of them, then you're rather severely immunocompromised. You're not likely to survive even a trivial infection. Also exciting was the fact that not only mammals and insects depended on this system to become aware of infection, but even plants did. That we didn't realize for a few months. We had to look at a lot of tear domain structures and use rather sophisticated search methods, but we found that there were the same conserved types of proteins even in plants. Some of these proteins are cytoplasmic, some are on the surface of plant cells, but if you mutate one of them, as I've shown here, you wind up with a flax plant, for example, that's susceptible to a fungal rust called Melampsora leani. This tells us that this system for host resistance is probably 1 billion years old or so. It goes back to the time before plants and animals split from each other. I was absolutely addicted to this positional cloning approach by that time. I loved it. The fact that one can start with a phenotype, make no guesses about the system at all, and wind up with a solid piece of knowledge like the receptor for recognizing microbes, that was intoxicating. And I wanted to do much more with it. I wanted to look at TLR signaling with it, but there weren't any other spontaneous mutations that we could use. And so in the year 2000, I moved to the Scripps Research Institute, and I set up a mutagenesis effort to create new phenotypes in mice, especially immune phenotypes, and then track them down. Of course, we kept many aspects of immunity under surveillance, not only innate immunity, and we've even become broader than that. But the idea was always start with phenotype, make no guesses, find the cause of an immune defect, and then you'll know something right away. Now this is done by injecting ENU into male mice, the G0 mice. They're bred to make G1 animals that are heterozygous for, we now know, about 60 to 70 mutations. But in the beginning it was a blind process. Those mice would be bred with ordinary females to make G2 daughters and then cross to their own daughters, whereon in the G3 generation we would have homozygosity for some of the mutations and we could detect recessive phenotypes. We know today that if you see a phenotype, it's almost without exception due to a change in coding sets, and you should keep that in mind. We only knew that the method was working because we saw a lot of strange-looking mice like these here, and we tracked all of those mutations down, but for a while it wasn't particularly easy. Before the mouse genome sequence was published, one had to build contigs, and even afterward one had at least some mapping work to do. And only with the introduction of ultra-high throughput sequencing methods in about 2009 could one rapidly track down a mutation in even a large critical region by sequencing the whole genome. So using various levels of difficulty, we tracked down 50 mutations in 26 genes that would affect TLR signaling. And these fell into proteins that acted as chaperones, or were the receptors themselves, or were adapter proteins, or kinases, or ubiquitin ligases, or transcription factors, or proteins that process TNF to allow it to be secreted. At all levels of the process, we found mutations, and we learned a great deal about how the signaling works. But by 2011, we were growing rather fatigued. we understood that the bottleneck in the process had to do with genetic mapping. The paradigm always was establish a homozygous stock of your mutant, cross it to another strain that has markers, then backcross, then genotype and phenotype the offspring, establish a critical region, then sequence the mouse. And that was too slow. Postdocs would declare phenotypes much faster than they could track them down, and we had a growing backlog of projects. Eventually, more than 100 phenotypes were sitting, waiting to be cloned, and this created an intolerable burden on our resources because we just couldn't store that many mice, and something faster was clearly needed. I envisioned a kind of magical method. Actually, this is a good way to go about solving things. Just dream of what you want, and even if it seems ridiculous at first blush, sometimes it turns out to be not so difficult. I thought we wanted a magical tool with which we could look at a family of mice like this one. And this tool would tell us which the affected mice were, even if it weren't so obvious as I'm showing you here. And not only that, but within the blink of an eye, it would say, this is a mutation in SOX10. And these are the coordinates of the mutation here within the genome. It's on chromosome 15. That's the protein change. This is the motif that's affected. And there is a human ortholog. And hey, here's the structure. And you even see the yellow amino acid there that's changed. And as I thought about this, I realized that actually we had all the tools to do this. They just had to be put together properly. So today we do that. Today we find mutations instantly. And by that I mean within 1 hour. Or else we declare that they're non-genetic in origin. So how is that done? Just like before, we mutagenize mice. We produce G1 animals. But the difference is that now we immediately sequence the whole exome of every G1 mouse. And that way we see all the mutations that they have and that they might transmit to the G2 and G3 generations. We know the number of mutations in general, and it has quite a bit of variance because we've sequenced thousands of exomes now. The mean number is 63, and that keeps fluctuating a little bit. But if there are more than 30 mutations, we deem the G1 worthy of investigation. We don't like to bother if there are fewer. We like to look at a large collection of mutations all at once. And if there are 30 mutations or more, we order an AmpliSeq panel. This is a collection of PCR primers that let us genotype every mutation site, all in a single reaction vessel. The primers take a while to arrive. Meanwhile, we make G2 daughters from that mouse. We make up to 50 G3 animals, and then we genotype all of them. And only then, when they're pre-genotyped, so we know the zygosity of every mutation in every mouse, then we distribute them to the screeners. The screeners look at about 110 different immune phenotypes sequentially. And these are usually quantitative estimates of antibody production, let's say, or how much cytokine is made in response to LPS. And we look at many other things, too, at glucose tolerance, at body weight, at certain neurobehavioral phenomena as well. The more we look at, the better, because we're able to solve the phenotype instantly. As of 5 days ago or so, we had generated 73,000 mutations. These numbers are exact, by the way. We know that 71% of all mouse genes had at least been damaged by mutation and then studied in this system I've outlined. This would be a huge burden of mutation. You can imagine no mouse would survive with 73,000 mutations peppering its genome, even in the heterozygous state, but they're distributed across 32,868 mice. We know that 19% of the genome has been mutated to phenovariants, and I'll explain exactly what I mean by that in just a little while, and examined 3 times or more in the homozygous state in 1 or more screens, and usually that means in all the screens. In adaptive immune screens alone, looking at the formation of lymphocytes and also their ability to make antibody responses, we've found mutations in 70 genes that were previously known to be involved in immunity, but also in hundreds of genes that were not known, which appear to cause phenotype. And that says that there's a lot that we don't know still. It's not a closed case. There's much to discover in immunity. We don't even know all the parts of the machine, or nearly all. We had to write software in order to look at these data, of course. And we wrote a console program called Linkage Explorer, and I'll just show you in a little bit blown up image of that, an example. Say we want to restrict our inquiry just to things having to do with CD8 cells, a kind of T lymphocyte. We can enter that string. We can say we only want to look at large pedigrees with at least 20 mice. We don't want to consider any mutations that haven't been tested 3 times or more in the homozygous state. We want only highly significant data, and then we click Submit. And we get back immediately a list of genes. Some of these genes, if any of you are immunologists, would be familiar to you, like Themis. This is a gene needed for CD8 development. Some would not be familiar, like SNRNP40, which is not known to be involved in immunity, yet as you'll see it is. In the next column you see the coordinates of the mutation, the predicted effects of the mutation, the screen in which the phenotype was observed. And if you go over to the right, you see a collection of p-values of the association between the phenotype and genotype in an additive level of inheritance. And what one can do is simply click on one of those numbers, and you immediately see a Manhattan plot. This shows all of the mutations organized chromosome by chromosome across the genome. Only one of them is significantly linked to this CD8 phenotype, a paucity of CD8 cells. The red line indicates the Bonferroni correction limit, and if you mouse over that point, you see what gene it is. It's the SNRNP40 from that pedigree. You may not know what SNRNP40 is, so you click again on the point, and the computer has already pre-computed for you what the gene is, what the protein looks like, what its domain structure is, what the mutation does to it. If you want to look at the gene model, you click again, and you see that here the mutation affects the critical splice junction and is predicted to make an in-frame deletion in the protein. You might want to look at the actual data, and then you right-click, and you see the phenotypic performance of homozygotes in dark green, heterozygotes in light green, and reference allele mice in yellow. And then at the extreme left are control black 6 mice. So this mutation apparently reduces the number of CD8 cells. It's not completely persuasive to me yet, and shouldn't be to you, because there aren't that many mice. And what we can do, fortunately, as we make mutations in the genome and they keep accumulating, is to look at multiple alleles. Gradually, we'll have mutations in all genes, and the computer recognizes this when it occurs and puts the data together into what we call super pedigrees. At present, 57% of genes in the mouse genome have been hit twice or more, and the super pedigrees give you greater confidence that there is a real association if it exists. So we can look with the same program at super pedigrees, essentially, and here you see the p-values are much, much higher. You can click on one of them, and you see essentially a spike where there are hundreds of mutations below the Bonferroni correction limit because they come from about 19 different pedigrees, and only one mutation, which is shared by all those pedigrees, has this extremely strong phenotypic signature. If you look at the raw data now, you see that there are lots of homozygous mice, and while there's overlap between the heterozygote and homozygote genotypes in phenotypic performance, still there's absolutely no question that this is the causative mutation. This is the type of mutation that would have been almost impossible to map by the old-style qualitative positional cloning. Yes, it's affected. No, it's not. You'd have a lot of doubt in your mind about many of these mice. But we've solved it kind of automatically. Super pedigrees can be looked at in different ways now. You can choose a phenotypic screen and say, I want to look at all the mutations that have super pedigrees that seem significant. This is hard for you to read, but we're looking across the whole genome, and you have the recessive model, the additive model, and the dominant model of inheritance, one on top of the other. And I blow it up for you here. We're looking here at the BT ratio as our phenotype. And you see these spikes that come above the Bonferroni correction line. This is all interactive. You can look at them the same way I showed you earlier, and you can expand the plot and you can see what all of the genes are that exceed the limit. You're also warned by a yellow color if this is likely to be false and attributable to linkage. Now, how much saturation have we achieved? There are many ways to define saturation, and I won't belabor them all today, but I'll just say we use mutation to phenovariance as our index. What does that mean? Well, we know that some genes are rendered functionally null by the mutations. That's the lower curve. Presently, about 7% of genes have been rendered functionally null. Premature stop codons or critical splicing errors will do that. And that's where we are with the red line, you see. We're at about 7% here, and as we mutagenize, the red line keeps moving that way. This is an overly conservative estimate of how much damage we've done, because we know very well that if you see a phenotype, it's twice as often due to a missense mutation as to a null allele, just because missense mutations are so common. You can take as a different estimator either probably null or probably damaging mutations as assessed by the program polyphen2 that predicts the damage of proteins on various bases. And yet we know that's too liberal an estimate because a lot of things that polyphen predicts turn out to be functionally neutral, about half of them in fact. And therefore we take the average between the two. We know that these curves bracket the amount of damage. We assume that the true amount is right in the center. What have we accomplished then? Well, in 5 years, it used to take us 5 years to find a mutation. Now, as I told you, it takes 1 hour. And in fact, the postdocs don't even really notice the mutation. The computer does in the majority of cases. So they need only look at the data afterward and decide which mutations are there. In the old times, we could only do one phenotype in 5 years. Now we typically solve 1 or 2 per day. So I say we're going 3,000 times as fast as we did a few years ago. We are limited by the rate at which we can put mutations into the mouse genome and by the rate at which we can screen mice. In fact, the latter rate is the most constraining now. The postdocs tell me they can only do 600 mice per week and that they'll die if they do any more. And I accept that because actually I know that is a lot of work. We can predict that within about 3 years we'll reach the 50% mark of mutation to phenovariance. And at that point we'll be able to say that we know most of the genes needed for immunity to operate as it does, or at least as we've defined it, according to the screens we're doing. But establishing mechanism is the real bottleneck. You can have a fantastic phenotype, and it can be in an exotic gene, and you may still have no inkling how that comes about. And that's what you're really called upon to do these days. Then again, I can tell you it's a lot better to have a problem with mechanism than to not know the cause of the phenotype at all, and to agonize over it for years before you even reach that milestone. So we're in a better place. One very quick example. Here we had a mutation that affected the inflammasome, one of the main arms of inflammation as we understand it, the NLRP3 inflammasome. You can see this mutation abolished inflammasome responses, and these are the homozygotes, heterozygotes, reference allele, and in the top you see the Manhattan plot. This was a mutation in NEC7, a kinase formerly associated with the cell cycle and specifically with the mitotic phase, with organization of the spindle and abscission of the cells after mitosis was complete. With only a very little bit of work, it was possible to find that NEC7 binds tightly to the NLRP3 protein, And it is, in fact, a critical component of the inflammasome. So we at least have a direction to go in understanding the structure of the inflammasome now. Previously, no one could crystallize the inflammasome. Maybe it might be possible once we know all the proteins involved, and it's opened the door to a lot of progress. In the early days, there were 4 leads on cloning the LPS project. Alexander Poltorak was at the top of the list, but Betsy Layton, Christoph van Uffel, and Irina Smirnova helped greatly too and stuck with the project for 5 long years. Nowadays, we have a group that does 5 different processes: making the mice, sequencing and genotyping them, screening them, doing the informatic analysis, and finally verifying the mutations by CRISPR targeting. That takes a lot more people. And these people, I hope they look very happy to you. They seem to me to be, because it's a compelling business, being able just to mutagenize and to expect with confidence that somewhere in the lab 1 or 2 fantastic phenotypes will pop up each day. And they have a lot to work with from this point forward. But they're great pioneers. They love the exploratory side of science, just as I do, as I think probably many of you do. We're explorers, just like the geographical explorers used to be. Before everything was found on the face of the earth, you can't say the same about the interior of a cell or how the immune system works. And these are wonderful times to be discovering these things. Thank you.
Muito obrigado, professor Beutler, pela maravilhosa palestra, e vamos abrir durante 20 minutos para questões do público e depois para a imprensa. Então, por favor, fiquem à vontade. Quem quiser fazer a pergunta em inglês, pode fazer, e quem quiser também fazer em português, pode fazer. Pode ser uma pergunta geral ou específica da ciência que ele apresentou, ou geral no que se diz respeito ao Prêmio Nobel, etc. Primeiro, eu queria pedir às pessoas que se apresentassem antes de fazer a pergunta, por favor.
Professor Butler, thank you so much for coming here and administrating the speech. And what? A little louder. Mais alto. Okay. Thank you so much, Professor Butler. I'm Arthur de Senha. I'm second-year medical student at this university. And I'm involved in a work in which we are trying to elucidate the role of the microbiota in modulating the immune response and therapeutic approaches related to it. You have talked about the C3H8EJ mice, which have lower sensibility to LPS. I have read that these mice also has a great vulnerability to Clostridium rodentius infection, which is similar to human pseudomembranous colitis. In clinic, we have tried to target these by fecal transplantation, in which we try to induce a protective microbiota into it. And we have had highly variable rates of success in this. My question for you is, could these highly varying rates of success be explained by innate differences in response to microbial antigens, and if so, how can it be applied to the clinic and the therapeutics? Thank you.
Thank you. That's a really interesting question and a complicated one, because of course you have a situation in these fecal transplant studies where you do have inter-individual genetic difference, and no doubt you are transferring different flora to people. Homeostasis is achieved in any individual based on a balance between innate immune potential and to some extent adaptive immune potential as well, and a particular set of flora. So it's easy to understand reasons why it might work in some people and not in others, just based on what flora are there, how much it's been depleted with antibiotics prior to development of pseudomembranous enterocolitis, what flora you are giving. And I can't give you a definitive answer, of course, but I can tell you those are the possibilities that immediately occur to me.
Hi, I'm Camila. I'm a PhD student at University of Católica here. And thank you so much for sharing with us your knowledge. And I would like to know from you, what do you think about IDR peptides as immunomodulatory molecules? We have been studying them not as antimicrobial peptides only, but also as immunomodulatory tentatives to help the organism to develop an infection. Thank you.
I see. To help the organism defend against infection or to develop infection? You're talking about a microbial peptide that would interact with the host?
Yes, I would like to know what do you think about them? These immunomodulatory molecules, they can really help the host to develop the infection caused by pathology.
I'm sure that such mechanisms exist in microbes. We know microbes have been fighting each other, first of all, for billions of years. And we know that they're very effective at it. And they've developed molecules to which resistance sometimes seems to be very difficult to acquire. It's also likely that they use peptide mechanisms, peptide-based mechanisms, to establish a foothold on mucosal surfaces, and that they change the characteristics of the host immune response. We could probably think of some examples of that where it clearly does occur, with fungal peptides, for example, that really interact with the cyclophilins, for example, or molecules that interact with cyclophilins and do change immune function. So definitely there's a lot to be discovered there, and actually it's something I wish I were working on.
So my name is Andre Nicola. I'm a physician and researcher as well. I'm more researcher than physician, I guess. I could relate with your life story. It's similar to mine, even though I'm at the very beginning. I have a specific question regarding the experiments. I've been studying for several years a specific immunological question, and I think the tools you developed and the mice you have and the data you have could actually help me answer my question. So the specific question is, how can anyone else from outside your lab have access to this data, or could we collaborate in some way to try and answer my question?
The answer is, first of all, you can see all the mutations we've made, and now there are about 300,000 of them, and you can have those mice if you want them, although we don't distribute them anymore. We're giving them all to a repository, so you'll have to get them from the MMRRC. But go to our website, mutagenetics.utsouthwestern.edu, and you can browse those mutations if there are particular genes you're interested in. Within about 6 months, we'll also make the phenotypic data available, so you'll be able to say, I'm interested in CD4 cells. I want to know all the genes where mutations affect CD4 cell numbers. And you'll be able to search for that too, but it's not quite ready yet.
Hi, my name is Elena. I'm finishing off the biological sciences undergraduate program here. And I'm sorry, I'm a little bit greedy. Can I ask 2 questions? So the first question is, in your professional opinion, is there any evidence that toll-like receptors also bind to self-associated molecular patterns? And do they in any way negatively regulate the innate immune system?
Well, negatively or positively. It could be, and I believe probably there are endogenous molecules that activate TLRs, to put it very quickly. I believe that most of those that have been identified as such so far are not too trustworthy. I would demand to see crystallographic evidence that there's really interaction and that it creates the conformational change known to be activating. I believe that in sterile inflammatory diseases, at least some of them, I'd be very surprised if it were not caused ultimately by activation of TLRs by endogenous molecules. And there might be things that act as antagonists too and that dampen the response. The only endogenous one I really trust is DNA at the moment, or RNA, because that's been quite well shown that not only microbial DNA is capable of activating TLRs. What's the second question?
Thank you. So my second question is completely unrelated. You, as a professional both of immunology and of doctoral sciences, do you feel like some scientists have recently said that we as immunologists mainly tend to focus on antibodies as a potential for therapeutic and clinical, while ignoring the innate immune system or cytotoxic T cells? Do you feel like all of our clinical research is mainly focused on antibodies while these other areas are being ignored? Or
I think that was the case before, but it couldn't be said to be true anymore given the big explosion of interest in innate immunity. Antibodies are wonderful, by the way. They really do have a great correlation with protection when you immunize, and they can be used for all kinds of things. They can be given passively to people rather than inducing them and so forth to treat inflammatory disease. But there is certainly a growing interest, and there will be a growing role of innate immune activators too.
Thank you so much.
Sure.
More questions? Okay, one more there.
I will take just one question. In this way of inflammatory disease, how to balance therapeutic to maybe blocking TLR receptors, but not make it so blocked all the time? How can we balance therapeutic and the necessity of response by our immunology?
Well, it's a good question. We have to see what happens in clinical trials, whether we create some real problem by blocking TLR signaling. Most of the drugs we use are sort of partial inhibitors. And even if you targeted all the TLRs by inhibiting MyD88 and TRIF, let's say, if you could do that, even though they're not particularly great targets, in general you wouldn't be 100% effective. And one can aim for drug concentration that's in the right therapeutic window and doesn't cause serious immunocompromise. There might be occasions where you want to totally block signaling by the TLR pathways. Maybe during sepsis that might be the thing to do. We're always saying, we MDs, that we can cope with infection, we just can't cope with the consequences of infection. If that's really true, then you might like to take full control of the situation and rely on your antibiotics alone to do the job of providing immunity while a patient becomes balanced again. But all these things are speculation, and they have to be sorted out by real prospective clinical trials.
Hi, thank you for talking. My name is Cecilia. I'm trying to do some research in immunology here. I try to do some interesting classes in immunology. My question to you is, how do you think about your data, all your data with these mice, and how you think about this to human? I think you may... This is not a new question for you, I think. How you think about this phenotyping and genotyping for human? How can we maybe apply this?
Are you asking maybe, could we do exactly the same thing with human populations that we do with the mice, with instant identification?
Applying this data, maybe you have this phenotyping in human.
Oh, yes.
You can search for human.
That is the hope. It is the case very frequently that the mutations we find in mice have exact counterparts in humans. You find humans with MIT88 deficiency, with IRAK4 deficiency, UNC93B deficiency, mutations in the I-kappa B kinase complex, and their phenotypes are remarkably similar to the mouse. About 98% of mouse genes have at least a human homolog, and vice versa. 80% have an ortholog, and vice versa. And there are situations where a mouse mutation is not predictive of a human phenotype, but actually it's less common than the opposite. So I view the mouse as a scout to finding human immune deficiency diseases. And if we had a comprehensive list of the genes important in the mouse, it would be a much simpler matter to understand human immune deficiency or human autoimmune diseases.
Thank you so much for coming. We're all very grateful.
A little louder. Yeah.
Okay.
Good.
Thank you so much for coming. We are all very grateful for your presence here. And I am Jaqueline. I am a second-year medicine school student. And my question is, in terms of inflammatory bowel disease, do you believe that the increase of gram-negative bacterial population is the main problem to be solved in terms of relieving the symptoms of this disease? Because you were talking very much about the LPS development, and what do you think about it? Would it be a nice start? Yes, a nice approach.
Well, here's what I think about IBD. We do have a screen where we give mice an injury to the colonic epithelium with a compound called dextran sodium sulfate. Normally, mice heal completely and quickly, but exceptional mutant mice cannot, and they develop a chronic severe colitis. I see no reason why in humans the situation might not be similar. We all have enteropathogenic E. coli infection from time to time. Most of us get well right away. A few of us may develop a severe chronic colitis. That may be what some IBD is about. Some of our mutations do correspond to rare familial causes of IBD in humans, and it is likely the case that there are hundreds of causes of inflammatory colitis. These are things that affect the ability of the epithelium to heal, affect the ability of the immune system to clear bugs once they get into the lamina propria, and affect the ability to secrete antimicrobial peptides. Many, many things can probably do it. I wouldn't concentrate just on the gram-negatives, but there may be explanations there too.
Ok, nós estamos quase terminando com as perguntas do público, depois temos ainda que passar para os jornalistas e o tempo está adiantado. Então nós vamos fazer o seguinte, a pergunta e depois o professor Paulo César também fará a última pergunta.
Good morning, Professor. Thank you so much for this opportunity. My name is Victoria. I'm a postdoctoral fellow here at the University of Brasilia. Actually, I'm not from the immunology field. From your talk, we were able to see how your success is due to great ideas, insights, teamwork, patience, and of course, lots of hard work. What would be your general advice or a take-home message for young scientists who pursue an academic career? Thank you so much.
Well, thank you. It's such an open-ended question. I could probably spend an hour giving you all kinds of advice, and some of it would be good and some not. But one last useful thing that I hope comes across. People often tell you you must have a hypothesis to do good research. And there is nothing wrong with hypotheses, but you have to be very disciplined about using them. The work that we do, to a large extent, is hypothesis-free. We make no guess about what might be involved in the immune system. We just go out and create exceptions to the norm, and then we find the cause, and then we know things that we didn't know before. If you use hypotheses, then by all means try to test them very rigorously, try to prove them wrong. The big problem with hypotheses is human beings like to be right rather than wrong. And I hear young people talk about how I proved the hypothesis. And you should know you never prove a hypothesis. You only fail to reject it. And you begin to develop an inferential model about how the system works. If you can use hypotheses with that level of rigor, then you're a good scientist. And then you're going to be successful.
Thank you so much.
Okay, thank you very much. I think it was Paulo's question too, and mine too. So, thank you very much. I would like to just invite journalists around here, please. So you can put some questions to Dr. Boyle. Where are you? Jornalistas? Sim, por favor.
Will this be in English or in Portuguese?
Okay. In English or in Portuguese?
No. I will try in English.
What? Okay, okay.
Hello.
Go on, please.
Okay. Thank you. Dr. Butler, thank you so much.
A little louder.
Okay. I'm a scientist journalist at Correio Brasiliense. The main... Thank you. It's better. Okay?
Try again.
Okay. It's a little question, and I would talk with your studies. Could you talk about the possibility of drugs more effective, and also about genetic mutation to improve our immunocells?
I didn't quite understand all of it. I'm very sorry. Try once again.
Tem tradutor, então você pode fazer em português, que acho que isso fica melhor, por favor.
Em português? Ok.
Professor, muito obrigada. Eu gostaria de entender um pouco mais como os seus estudos poderiam trazer uma possibilidade de drogas mais efetivas e mesmo de mutações gênicas para melhorar e aperfeiçoar nossas células imunológicas.
Okay, very well understood now.
I'm so sorry for my English.
No problem. There are several possible fallouts from the work that we have done. On the one hand, we know that activating the innate immune system can lead to activation of an adaptive immune response as well. In the past, rather undefined methods have been used to help drive an adaptive response. When you're given a flu shot, for example, you're given a compound called alum, aluminum hydroxide usually, or some other aluminum-containing compound, and that creates inflammation, and we still don't understand how. Now we know certain receptors that do drive inflammation, and it's likely that one could develop molecular adjuvants with a known mechanism of action that are very effective in producing antibodies against whatever peptide we wish to give, or whatever protein or whatever organism we wish to target. So vaccines may get better. It may be also that we discover the fundamental cause of some inflammatory diseases that are a big problem in medicine. Rheumatoid arthritis, systemic lupus erythematosus, ankylosing spondylitis. In some of these diseases, we know that TNF is produced in abundance, and yet we don't know why. We don't know what's driving it. Perhaps it's receptors of this family that I've been talking about, perhaps other innate immune receptors, but there's the potential that we could have very specific therapy for that kind of disease. The work that we've been describing recently, the rapid identification of new mutations that affect immunity, it may have a big effect also on human health in that many immune deficiencies are not understood genetically. If we can find the cause in the mouse, then it may be very easy to find humans who are just like those mice, and to have a model available immediately for study of therapy.
Hi, I'm Carolina Samorano from Metrópolis website. Eu vou seguir a linha da minha colega e fazer a pergunta em português. Sorry. Para evitar mal-entendidos. Uma pergunta um pouco menos técnica. Eu queria saber, o senhor comentou algumas vezes durante a palestra sobre o papel da tecnologia e como a tecnologia permitiu que os resultados alcançados fossem mais rápidos e tal. E eu queria saber a que a ciência deve, o que a ciência deve à tecnologia hoje em dia, se existem pesquisas, por exemplo, que talvez a gente não teria conseguido fazer ou ter resultados, enfim, como a tecnologia, todos esses anos que o senhor atua como pesquisador, fomentou o avanço e o alcance de resultados.
I would have to say that the great majority of the work we do now wouldn't be possible at all without the recent technological progress that we've seen. And this applies at every level. If we're talking about DNA sequencing, a great deal of engineering, chemical and physical engineering, went into designing the current generation of sequencers, which let us cover the genome, developing sequence about 10 million times faster and 10 million times cheaper than was the case 20 years ago. If we're talking about interpreting those results, then the computational power available to us now is hugely increased, and again, we couldn't have gotten very far a short time ago. There are other new technologies that we use to verify our results. You've probably heard of the CRISPR-Cas9 method for targeting genes. It's a simple matter these days to inject certain short guide RNA molecules and a certain enzyme into cells and target a specific gene and even replace a single nucleotide. We've done this with close to 100 targets in the past year and a half or so, and we probably would have only been able to do one by the previous technology. I'm frankly amazed at what's possible now. If you had told me what we would be able to do, that we could clone mice, transduce cells, make targeting of single nucleotides, if you told me that 30 years ago, I would have thought that's crazy. It'll never be possible. And yet it's routine now. And one can only imagine what will be possible 30 years from now.
Uma outra questão, Dr. Butler, seria, de acordo com as suas observações dos últimos 15 anos, de sua observação, qual seria o principal ator do nosso sistema de defesa? Seriam as nossas próprias células e a adaptação delas às bactérias, ou seriam os antibióticos, a ação dos antibióticos e outras drogas?
It's an interesting question, and it calls almost for a little bit of philosophy to answer it. When we had no antibiotics and no vaccines, let's go back just 150 years or so to the Victorian era, to an industrialized country, namely the United Kingdom. In those days, the mean lifespan was about 30 years or so, and median lifespan was about 10 years. That was what a child could expect from the date of birth. Most children died of infection by 10 years or shortly thereafter. Antibiotics, sanitation, vaccination have all changed this enormously, and in England today, the median lifespan is about 8 times longer. So it's almost as though we're a different species with this little bit of assistance from antibiotics and various forms of ingenuity. At the same time, if you had no immunity, that would be of no help whatsoever. You wouldn't ever make it past your first few weeks of age, even with assistance from antibiotics. So immunity is the fundament. Innate immunity is probably marginally more important to us than adaptive. You can live without an adaptive immune system or with a very severely impaired one for a period of months or so, perhaps. If you had no innate immunity, if you even lack neutrophils, let's say, you're likely to get a severe infection and die within a few weeks. Immunity is more important than anything we've come up with. That's the answer. But what we've come up with has had a dramatic effect on human health and on the whole quality of life and just what humans are able to do with their lives.
Okay, I have another question. Falando em antibióticos, eu obviamente não sou médica, mas eu sei que a gente está numa era em que a gente se discute o uso exagerado de antibióticos e o que isso pode trazer de consequências para a nossa saúde, seja por desenvolvimento de superbactérias, enfim. Até que ponto o desenvolvimento, as pesquisas para que se chegue a novos medicamentos e novos antibióticos podem, assim, existe um embate científico quanto ao antibiótico, até que ponto ele é uma ajuda, óbvio que é importante, mas até que ponto ele é importante para nossas defesas, se isso não está matando o nosso sistema imune que já vem de fábrica. Gente. Thank you.
Definitely the antibiotics aren't killing the innate immune system we're born with, and you have to realize antibiotics have been present only for about 70 years. In all of human history, we've only been protected by antibiotics for about 70 years, which is not enough time for it to have any appreciable effect on our evolution as a species. It hasn't weakened us appreciably. What is true is that antibiotic resistance is developing and has become a major problem, and there are many types of bacteria that used to be easily dealt with antibiotics that no longer are. We might expect that the next generation and the one after might not have the same protection we have, we who have lived in the golden age of antibiotics. There's nothing to say that antibiotics will be good for another 70 years or for another 200 years. It will take probably more intelligent use and management of the use of antibiotics than is going on at present if we're to have any chance of that, and it will take more aggressive development of new antibiotics. That, by the way, has been quite a problem. They're not being developed as quickly as most people would like. And nobody even knows whether it's possible to keep ahead of the microbes by means of human ingenuity.
Okay, thank you. I think we can stop here. Would you like to say something else, or just? I think our immune system needs some food, of course.
Well, I just want to say thank you again for this huge honor.
It's us. Thank you very much, Dr. Boite.
Thank you.
Very nice.
Thank you.
For our presence here in the university. Great.
Thank
Great
you.
for us.
Well, it's wonderful to be here. It really is. I like Brazil.
That was a good question for our students.
They were good questions.
I'm an engineer and it was very difficult. Sure.
Thank you very much.
Oh,
Thank you. Thanks a lot. Did you get the gist of it as an engineer?
Excuse me?
Did you understand it a little bit?
It was very hard.
Oh, well, I'll give the engineer's version next time. But engineers make everything possible.
Yeah, you good?
Yeah.
You're free.
I'll let you sort that out.
Thank you.
Thank you.
you talk about this nuclear factor capability. I read about some articles that you could downregulate it with because of nitroic acid or upregulated with arachidonic acid. Do you think that this could be an answer to controlling this disease or the cause of this disease, the high consumption of arachidonic acid, for instance?
I don't know.
Okay.
You know, people often talk about hydrogen hypothesis, but I think there's so many things changed in the modern world. It doesn't have to be worms or hydrogen. It could be a lot of things.
Thank you very much. Okay. Would you mind taking a picture?
Sure.
Sorry, I have to take you away. I do. That was great.
I'm sorry.
I'm really sorry.
How about all together?
Okay, that's a good idea. Everybody together.
They all want their own phones, of course.
Sorry, do you mind? Thank you so much.
Okay.
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 Thank you.
Thank you.